Good morning, everyone. I am LEBSIR Dalila, a lecturer at Jijel University. First of all, I want to thank the community organization for allowing me to present this work about impact of repeated potassium iodide intake in case of protected radioiodine release. The context is about nuclear accident and how to manage the risk. Well, the most severe health consequence of nuclear reactor emergency with the release of large amount of radioiodine, such iodine 131, is the development of thyroid cancer, especially among young population. As demonstrated in this graph, in Ukraine and Belarus, year after Chernobyl disaster, we have a huge amount of incidence of thyroid cancer. To counter this threat, competent authorities implement series of countermeasures. We have physical approach like food restriction and evacuation, and pharmacological approach with a single administration of potassium iodide at 130 mg. This to block radioiodine thyroid uptake. This approach is very efficient if applied few hours before exposure to radioactive iodine, taking into account that a high dose of iodine transiently impairs thyroid functioning. The well-known Volchenkov effect. Before talking about the aim of this presentation, I will take a few minutes to explain the thyroid hormone synthesis and the famous Volchenkov effect. As we know, iodine is an important trace element for the functioning of the body. As our body doesn't synthesize this element, the required daily intake of iodine should be around 150 micrograms. Thyroid hormone synthesis requires a series of regulated steps. Before starting, all the abbreviations are described in the right upper corner. So, firstly, NIS actively transports iodide into the thyroid. Then, iodide efflux into the follicular lumen is mediated by pendrin. In second part, at the interluminal side, iodide is oxidized, a reaction that occurs hydrogen peroxide. The oxidation of iodide is mediated by thyroid peroxidase, TPU. This TPU is also responsible for the iodination of selected tyrosine residue of thyroglobulin, forming monoiodotyrosine, MIT, and diiodotyrosine, DIT. And after that, for the coupling of this element, resulting in the formation of thyroid hormone, diiodotyrosine, T3, and tyrosine, T4. Finally, thyroid hormones are released into the bloodstream to transporter monocarboxylate 8, MCT8, following pinocytosis and lysosomal digestion of thyroglobulin. Thyroid hormones exert a major impact on development, growth, and metabolism. But what happens in situation of iodide excess? Well, in this case, the thyroid is switched off. We get transient inhibition of the main steps of thyroid hormone synthesis. We call this adaptive phenomenon Volchenkov effect. 24 to 48 hours, we usually get an escape from this Volchenkov effect, and thyroid function resumes. If the escape doesn't happen for any reason, then we are faced to a big problem. The individual could develop a hypothyroidism. So, what Fukushima MPP accident teach us? Well, this accident raised many questions about how should we proceed in case of prolonged exposure to radioactive iodine. You know, many episodes of this continuous radio element release over several days happened during Fukushima MPP accident. Such situation requires more than one dose of potassium iodide. However, knowledge about the effect of repeated administration of KI in sensitive populations remains scarce. So, the aim of this work was to evaluate the effect of repeat administration of target dose of KI, potassium iodide, in pregnant rat model, mimicking a sensitive model, the progeny. Well, the present slide is a scheme of a human brain development in relation with thyroid hormone secretion. As we can see, thyroid hormone receptors are present since week 10, whereas fetal thyroid hormone secretion takes place around week 20. During this early fetal period, the development of many brain structures is under maternal thyroid hormone control. As we saw previously, the variation of iodine may lead to a variation of thyroid hormone level. You know, the deficiency of thyroid hormone during fetal development may lead to several syndromes, and classically we will distinguish neurological or mixed matters cretinism, which is a condition of several stunted physical and mental growth owing to untreated congenital deficiency of thyroid hormone, usually owing to maternal hypothyroidism. To assess the effect of repeated intake of KI on the central nervous system of progeny, pregnant rats were given KI since gestational day 9 over 8 days, and then 30 days after, the weaning offsprings were subjected to behavior tests. After that, thyroid and brain were collected to study the impact of KI exposure in these organs. The first result demonstrates a significant impact of repeated treatment on thyroid hormone level. We observe a decrease of free thyroxine level in conjunction with decrease of TSH level. This result is quite uncommon, since thyroid hormones exert a negative feedback on TSH. In the literature, this phenomenon is normally due to an impairment of pituitary gland. To assess motor activity, we used a rot test. The principle of this test is that rats are first trained to walk on a rod rotating at certain speed. Animals experiencing impaired motor coordination are unable to cope with the rotating rod and will drop off when the rotating speed exceeds their motor coordination capacity. In our experiment, we observe a significant impact of repeated treatment on motor coordination, as the time spent on the rotating cylinder is significantly decreased in treated groups. At the molecular level, we obtain a significant decrease of cerebrome genes expression involved in granular cell migration and synaptic plasticity. On the other hand, we obtain a significant increase of cortex gene expression involved in myelination and neuronal plasticity. All these modifications led to a brain impairment and an asynchronization. To conclude, KI at 1 mg per kilogram over 8 days induce a neurotoxicity and motor impairment in male progeny. At the end, I want really to thank you for your attention.